Tomi, it's great to see you again, and to start a series of podcasts that I think would be very interesting for people in the peptide discovery field. And maybe we'll start with introductions. I have to say that, first of all, you are now in the position of the head of the scientific board in Pepticom, and we are extremely happy to have you leading our scientific advisory board. And we are working with you for many years now and learning every day from the work with you. So you are one of the key opinion leaders worldwide in the field of peptide therapeutics.
Thank you, Immanuel. It's a real pleasure to have had the opportunity to work with you and the team at Pepticom and to take upon the challenge of chairing this illustrious scientific advisory board. So thank you again for that. And in terms of my own background, I'm fairly well known in the peptide field because I've been at it for such a long time, both in biotech and pharma for 40 years as of now. Currently, I'm doing consulting and advisory work, obviously, and having the opportunity to work with you and the team. But throughout the years, and this kind of captured on this slide here, I've had many experiences working on different targets, ranging from receptors to enzymes to intracellular targets, protein-protein interactions, DNA and RNA regulatory targets. So it's been quite an excursion through target space, but also chemical space from the standpoint of structural biology, as indicated by some of the inserted 3D structures on the slide here. And with respect to a number of lead molecules that I've had the opportunity to lead teams to push these through early drug discovery, preclinical, and in a few cases, all the way into the market. One thing I would say is that I'm rather agnostic in terms of the modality itself, although the strength of my experience is obviously in the peptide field, including Peptidomimetics and Proteomimetics or Proteomimetics. One of the favorite things I've had the chance to do from time to time is de novo design. We'll be talking about that a little bit later in terms of nonpeptides, as well as outside of peptide space, natural products, macrocycles, and small molecules. My education started with work at the University of Arizona in terms of my graduate studies, and we'll actually talk about some work that's still ongoing in that regard on the melanocortins. This has been really a long-term endeavor, and I would say probably in some regards quite successful. It's led to hundreds of publications and patents as well as presentations throughout the last 40 years. So I'll leave that to be my scientific biography on one slide, and look forward to our podcast here.
Thank you, Tomi. First of all, you're extremely modest, so you have more than 600 publications, and as far as the melanocortin receptor that we'll probably review, did really outstanding breakthroughs in the field, so I'm very excited to hear more about that. I did a PhD in biochemistry and a postdoc, mainly in molecular biology. And then I left academia after publishing quite a few nice papers and basically started Pepticom together with a group of very talented people, most of them computational chemists. Dr. Amit Michaeli, our CTO, is basically a person that started and built the technology of trying to discover or design peptides from scratch using AI, and that's basically what we do in Pepticom. Amit did his studies with Professor Amiram Goldblum, which is one of the the founding father of computational chemistry in Israel. And that's the link to try and discover in a much more efficient way future of peptide therapeutics. And just to give a context for what we are doing in Pepticom and why this is relevant. So when we were discussing about doing a podcast, we both agreed that there should be a place where people hear a broad review about the field of peptide therapeutics and how it actually developed. I personally think this can be very educational because there's such a history to the field, and it's extremely interesting actually the way peptide-based drugs were discovered and developed, some of them derivatives of natural peptides, some of them completely novel. And I think it could be also great fun for people that are interested in science.
I completely agree. And perhaps I go to the next slide here, just because it does provide sort of a historical account. It actually goes back before I was even born. So that's the best part of it. And Yes, for sure. Back last century, which, probably for many people who are hopefully going to be listening to this podcast and have had experience in the peptide field will appreciate some of the work that was done before 2000. Most of the pioneering concepts, technologies, and also I would say the challenges that were encountered to develop peptide drugs were first realized back in the late 1900s, especially I would say in the 1970s, which led to what I call the first wave of peptide drug discovery. It's illustrated by the kind of the green circles and focused primarily on protein-coupled receptors such as for somatostatin or the melanocortins or GnRH or the opioids, as well as extracellular protein targets such as renin, a protease. And before that, I would say it's also really important to point out that some of the key advances in chemistry and biology, inclusive of structural biology, were achieved even earlier in the 50s and into the 60s with some significant first-time discoveries, including the alpha helix and the beta sheet and protein structures in general, and understanding the nature of amino acids from a structural standpoint. And making peptides, not an easy thing to do. Actually, just making peptides, starting out solution phase and then solid phase. And what always strikes me as remarkable was the synthesis of ribonuclease, both A and S forms, by Merrifield and the Merck team using both solid phase synthesis and solution phase synthesis. This was 120 amino acids, work done in the 1960s, truly remarkable.
I agree, and just looking at this time scale is really impressive because, first of all, for people that don't know, we were just finishing to celebrate the 100th year of insulin as a used drug, which is outstanding and a great achievement to human health. Everything that you just mentioned, all of this was done before the era of molecular biology, which is really outstanding, and all of these drugs were developed with technologies that are basically, we tend to see them today as obsolete, even though they are not. I mean, they are extremely important. If I understand correctly, all of these drugs are basically, in the first wave, are derivatives of natural peptides, right?
Yes, and a very good point about the molecular biology revolution, which was kicked off with the sequencing back around the year 2000. So both the first wave focused primarily on receptors and extracellular targets, and the second wave focused on intracellular targets, which is truly a major effort nowadays to develop peptides that are permeable to get into cells. All of that work, as well as even into the third wave of using diversity relative to synthetic or biosynthetic methods, from phage to both linear and cyclic phage display, DNA-encoded libraries, mRNA display libraries, as well as synthetic approaches for linears and cyclic, including cell-penetrating peptides. All of this work, all being achieved even before the turn of the century or the new millennium. And so in terms of the concepts and the technologies that I was mentioning a couple of minutes ago, all we are doing is really building upon those achievements and those understandings. And from time to time, like in other areas of science, we come across some dogmas that we think are correct, and we find out that they're not necessarily true. We just were limited in our understanding and or our tools used to study them. But I would say that, in terms of the nature of peptide drug discovery, these are the best of times we're in now, obviously because we have the collected wisdom of previous decades and achievements and failures. And now we understand what made both achievements and what made those failures happen.
If you touched on that already, what do you think was the biggest limiting factor in the past for discovery of, let's say, novel peptide-based therapeutics?
I guess, when you've been in the bunker like I have for so many decades, you kind of forget about the fact of where you've been and where you're going. But of course, the original work for decades up to the turn of the century was looking at natural sequences, oftentimes peptide hormones or peptide neurotransmitters, and then beginning to segue into protein motifs that were involved in protein-protein interactions in various ways. So the early work was really focused, I would say, on the peptide or protein motif itself and what people were hypothesizing if there was not structural information from X-ray crystallography or NMR, but hypothesizing whether they were helical or reverse turn or extended conformation, beta strand-like and so forth. And a lot of chemistries were developed in order to try to begin to bias peptide analogs thereof that were assuming helical or assuming reverse turn or assuming extended backbone conformations. Some really nice chemistries that have in some ways kind of come and gone by virtue of using macrocyclization chemistry, which we'll be talking about a little bit in this podcast. In terms of the way then and the way now is, I think, both structural biology and computational methods. There's no doubt that seeing how peptides are binding to their target and now receptors as well as proteases and protein-protein interactions, even DNA, mRNA interactions, all playing significantly into new ways to design and advance and lead optimize peptide drugs. The old way was looking at the peptide very myopically, and now it is looking at peptide target interactions, but also even just the target itself to do what I call in the past and still relevant now is de novo design, looking at the target, looking at the shape of the binding site, the nooks and the crannies, the surface, the complementarity to a ligand to bind to it, and coming up with a novel peptide which does not have to begin with natural amino acids if you don't want it to, and can include other kinds of building blocks. So to me, this is the exciting part of what I would call the fourth wave. And I think when we kind of close up this podcast, we'll kind of come back to what the fourth wave may be.
Yes, that would be extremely exciting because obviously that's exactly what we were trying to do in Pepticom. And I think that before we get there, it would be very interesting to see how it was done in the past. And so basically the first wave of peptide-based drugs, it's really interesting to think about that. They were all developed where, tools that we take for granted today for visualization and understanding of structure and structure activity were just not there. It's really disturbing to think about that, how difficult it was probably.
It does take me back a ways, but when I was learning about peptide chemistry, working at the University of Arizona with Victor Ruby, who was very famous in his own right, for sure, we had to make our own resins. We had to protect and make our own amino acid building blocks that we were using. We didn't have computational tools. We didn't have computers for anything, any purpose whatsoever. We had no internet. Everything was seemed to be, at the time, pretty high technology. Relative to now, it was very primitive. But I think in some ways, and if you look at the history of science, it's remarkable that scientists were able to achieve what they did before having all these tools in hand. So every time is exciting. When you're learning and contributing as a scientist, that's what makes it a wondrous profession. Yeah, it was difficult and it was slow. And nowadays, everything needs to be fast. To be competitive, you need to be fast. And I think that some tools like screening experimentally or computationally and doing it with high fidelity is really critically important. And that's the nature of the game now. It wasn't back in the 70s and 80s. It was basically, can you make it or can you not? Or can you test it or can you not? And just cell biology in itself was just beginning to evolve during that time. So it's been a continuous evolution of both chemistry and biology and all the interfaces between, including structural biology and computational. But it's nice to reflect upon it. So when we're talking about some of the earliest peptide drugs, there are many of them. They are certainly not all exemplified on this slide here. But some important nuances, I would say, are meant to be illustrated here. And that is some of the major contributions of peptides are really yet to be fully realized in terms of the marketplace. And as you'll see the text at the bottom of the slide here, of the global pharmaceutical market, peptide drugs are constituting about 5% at the moment. But that number is going to change quickly because there's some blockbuster peptide drugs forthcoming, especially for obesity and metabolic disorders. And these include the GLP-1 agonists and now a new series of GLP-1 agonists that are hybridized to be co-agonists with GIP and or glucagon. Also, insulin contributing to this in terms of treatment of diabetes. So just in two peptides, GLP-1 and insulin, a significant proportion of the peptide part of the global pharmaceutical market exists. So there are tremendous opportunities for other peptides, as illustrated here, including the melanocortins and the somatostatins and PTH, and yet others that are not shown on the slide here, all making headway, if not in the marketplace already, but into the clinic. And this is what is providing, I think, to me, great momentum. And even those who sit and evaluate trends in the global pharmaceutical market will say peptides are the up-and-coming newbies. Still oldies, but newbies in terms of making a significant breakthrough to expand in the therapeutic space and to expand in the marketplace.
I completely agree. And I think there's also something very important that we should think about. Most of the drugs, or all the drugs that you mentioned, they are basically derivatives and improvements on natural hormones. And this is growing the market significantly. And I'm imagining the impact that discovery and methods of completely de novo peptides are going to have in the near and far future. And they are probably going to really change and increase the size and the percentage of the peptides in the global pharmaceutical market. I'm convinced they will have a great impact. So just because they are opening a completely new possibility of designing peptide modalities that was just impossible to do a few years ago. So we'll probably see a few examples of how this is already happening, and I'm sure that it's going to increase dramatically in the future. It's going to be very exciting for peptide-based drugs in the future, I think.
Absolutely. And the more we understand about the biology about peptides, whether they're for receptors or intracellular targets, and how unique the triggering mechanisms may be, or if they're part of a cascade of parallel mechanisms, the sky's the limit. You can focus on novelty of peptides in terms of introducing the potential for oral bioavailability, which has always been a huge challenge in terms of anything that's a molecular weight of 1,000 or more, but that's changing now with respect to the field of drug delivery itself and new formulations and methodology and learnings, as well as the peptide component to it, and dealing with larger size or macrocyclization versus linear, and also understanding that once you get into the bloodstream, whether it's oral or whether it's IV or sub-Q or other nasal or whatever, to have the species that's getting into the blood also have good pharmacokinetic properties. So it's multi-dimensional opportunity. It's not just novelty in structure, it's novelty in properties. And I think this is finally, I can say after being in this game for 40 years, all of the necessary knowledge and databases and concepts and methodology tools are all in place to orchestrate, which is kind of what I'm doing in my consulting work. That's why my little business is called Maestro Therapeutics, because all I'm doing is trying to orchestrate the knowledge and skills that I have to support biotech, to help the peptide field. It's important to me. It's kind of what I hope to contribute as my own legacy, not just for myself, for the field. That's the fun part. That's why we try to have fun in our profession. And I've had plenty of it, plenty of tough times too, but to be honest, mostly based on natural ligands, mostly linear peptides is where we begin. But over the years and through the discovery of cyclic peptides, both from our own physiology, like the neurohypophysial hormones or insulin, or from natural products, especially those that may have antibiotic properties, we see that there are many macrocycles that have been around for a long time, more recently made it into the marketplace. I think that there's no question, if you were to ask most drug discovery scientists now in biotech and pharma and academia, Are macrocycles the hot thing? Yes, they are. And there's a reason for it. They also provide opportunity to potentially take advantage of their more stable structure metabolically, but also for oral. So even despite having a higher molecular weight, they may have a chance of being more orally bioavailable. And I think everyone believes that's the case. There's evidence now published over the last few years that supports that significantly, not necessarily with high quantitation, but with clarity that the approach works and that it's, again, as I said before, it's not only the peptide, but that has a lot to do with it. It's also the formulation. So I think we all appreciate that drug delivery isn't a myopic science either. It's multidimensional. And I think that's where we're going. And hopefully, as we progress into the future, there'll be more examples of the route of delivery being more oral or peroral.
We'll probably touch on that later on, but I think the near future is going to be extremely exciting in this regard with peptides that are in late clinical development right now.
And a lot of credit goes to Novo and Nordisk here, although there are many other players in this space. So I don't want to sound like I'm rooting only for one company here, but Novo has done some very nice work, especially to introduce the albumin binding modifications of the GLP-1 sequences, as illustrated by liraglutide and semaglutide, active ingredient now even in the oral form of GLP-1 from Novo. Important here also is this was a long peptide, 30 or so approximate amino acids in length. So this was a challenge. And Novo has gone about this in a semi-synthetic way to then incorporate through a single lysine residue side chain in each of these two sequences a fatty acid modification and variation of that in terms of linker and the fatty acid itself. So very important concept to the whole field. Not just GLP-1, but the ability to, what some people call hitchhike on albumin to improve pharmacokinetic properties, half-life, and exposure or concentration levels to achieve therapeutic activity over a period of time. And hopefully, in most cases, like for sub-Q administration, what used to be felt to be an achievement for a once a day now is transcended to once a week or even potentially once a month. A significant achievement to the field. And in terms of what came a little bit later relative to the chemistry and the biology work on the GLP-1 family has been getting a structure. This work also came from Novo, as shown on the slide here on the right, an X-ray structure of the GLP-1 receptor with GLP-1 itself, with the blue helix being the GLP-1. So we're learning as we go, and there was a time that was multiples of decades where we didn't even know what a GPCR really looked like other than anticipation that it was a multi-helical transmembrane domain receptor. But to understand exactly how the peptides were binding to it, let alone how they're transducing signal from the GPCR itself to adenylyl cyclase was another thing. The additional knowledge of knowing the structure of the target.
I mean, when was the first GPCR crystal made? I think it was a decade ago, or less than a decade, right?
I believe that's the case. And X-rays were one way to resolve structures. Cryo-EM now is another way, especially for those that involve lipid interactions. I think that we're seeing a significant growth of target proteins, receptors, catalytic proteins, non-catalytic proteins in the protein data bank and other repositories reflecting the significant momentum of structural biology relevant for peptides, as well as those that are trying to achieve similar effects with small molecules and peptidomimetics in between the two. So
I have to mention also AlphaFold, which is a great addition that was made like a breakthrough, I think 2 years ago, which would also have an impact. I think it's a very interesting technology. But going back to GLP-1, I think it's an excellent example of modifying a natural peptide. It's probably one of the best examples, right?
Yes, I think because first of all, it's a longer peptide. It achieved amazing impact to the field of diabetes first, as well as now to obesity with extraordinary impact for those who significant weight issues, give millions of people hope. And like I say, the sky's the limit on that one. Metabolic disorders have been not fully resolved, of course, with just GLP-1 itself, but it probably is the most significant impact in the field for a long, long time. So lots of excitement. And I think it's bringing the microscope back onto peptide drug discovery, that one particular example, which is something that we want to take advantage of. So the melanocortin also, at least one of the subtype receptors, the MC4, has also in relationship to obesity. And MC4 agonists are considered to be also part of the exquisite mechanism that controls feeding behavior. And in this case, an agonist would decrease feeding behavior, feeding drive, and also have application as a therapeutic for obesity. The work on melanocortins goes back a long ways, back probably to the 1950s, where the pigmentary receptor for alpha-MSH, what we now call the melanocortin 1 receptor, or MC1R, was discovered. A major contributor to the field academically was the group at Arizona, led by Mac Hadley and Victor Ruby on biology and chemistry. And I came into play in the mid-70s in graduate school to make a couple of analogs, which have marched along now both individually and as a starter, if you will, to marketed drugs. The first was the molecule shown here at the top left, SNES, or norleucine 4-DP7 alpha-MSH. Again, the natural sequence, just two modifications to make a peptide that now has been marketed for quite some time for the treatment of certain pigmentary disorders, especially for children, that allows them to be in the sun. Otherwise, they were suffering from a debilitating disorder that forced them to be indoors all the time. So this peptide also has a greater impact, obviously, for skin tanning and is being used in that regard as well. We hypothesized why a D-phenylalanine might be contributing to significant potency enhancement of 10 to 100-fold of this molecule SNES versus alpha-MSH. We conceptualized that perhaps it was a reverse turn, as illustrated in the left-hand part of the slide here, where we made a cyclic analog replacing the norleucine or a methionine in the original sequence and the glycine on the other side, positions 4 and 10 respectively, by a cysteine. This allowed then a cyclic or macrocycle analog that was tested and found also to be a superagonist, likewise to the D-Phe substituted alpha-MSH. So this one we refer to as cyclo-cis410 became also another benchmark in being able to transition from linear to cyclic in a logical way, if you will, hypothesizing on the bioactive conformation bound to the receptor. Of course, then and for decades to follow, there was no structural biology on these GPCRs, so it was all based on the peptide itself. Now, most recently, Rhythm has...
Before you go to the more recent history, so this work from 82, it's, if I understand correctly, one of the first works on real drug discovery efforts on cyclic peptides that you basically initiated then, right?
Yeah, yes, it was. And thank you for reminding me that it was a big breakthrough, at least within the peptide community, which was just beginning to really build worldwide. And I remember presenting this at an American Peptide Symposium way back then, probably was 83, and people were amazed. It really did create a tremendous incentive for peptide chemists to hypothesize on their own targets of interest and their own research cyclization, which led to even more and more examples for different GPCR peptides in particular, for which they were binding in kind of reverse turn conformation. Now, that was a subset, but a big one, of the GPCRs. The ones that include GLP-1, for example, were a different subset. We call them GPCRs Class A and Class B. The Class B would be the GLP-1 type of peptides. All were predominantly, and based on structural biology now, all helical. So you have two major secondary structures binding of peptides binding to GPCRs, reverse turn such as the melanocortins, and helical, as exemplified by GLP-1. So in retrospect, really profoundly interesting and high impact to the field, as you mentioned, Immanuel. Relative to biotech taking advantage of the cyclization that I introduced back in the early 80s, made the analog shown here, setmelanotide, which is now marketed for genetic disorders involving obesity. And setmelanotide obviously is very similar to the cis4 cis10 alpha MSH and includes a couple of modifications, but otherwise is extremely similar to the original peptide that I made. More profoundly of impact is the cryo-EM structure of setmelanotide bound to MC4, which underscores the fact that we had a pretty good idea a long time ago. It actually does bind in a reverse turn conformation, and although a nuance to it, but it does also incorporate a calcium ion interaction, which was known also biologically to be fundamentally important for the melanocortin signal transduction. So now we see calcium bound between the peptide to the GPCR active site, and I can say it all makes sense now.
It's amazing to see how the structural knowledge you were able to actually make a cyclic peptide that had such a potent activity.
Well, my PhD advisor, Victor Ruby, favorite quotes that he used to share with the group when I was there was from Linus Pauling, and it goes something to the effect, to have a great idea, you need to have lots of ideas. One of them will actually work out. But this was a good example of...
It's a very good quote.
Yeah, it's not really I did a poor job to reiterate it, but it's really true. These days, I guess you could say, you can use technology to test your many ideas. Back in those days, you had to make a decision on which of your ideas you want to actually work on because things were very slow process. But this was one that worked out. I did make some other cyclics, and they were not as good, but they did work, but they weren't super agonist like this one was.
Yeah, just a take on what you just said. I think that we are now approaching a stage in history, time in history, where technology can not only help you in testing your ideas, it can also help you in generating ideas. And I think that this is something that would have a great impact on the future of peptide drug discovery, because think about a few chemists sitting all day and trying to solve or design a peptide. If you could at least some of this activity perform on a computer on large scale, that would save years of work, peptide chemistry.
Yeah, that's exactly right. And I think this, you provided the segue to jump back to the waves of peptide drug discovery, and that does constitute the fourth wave. We've gone through receptors, we've gone through intracellular targets as well, we've gone through diversity by various methods, and now we're into using the sophisticated computational, and I would say that it integrates, with human mind. The computers are only as smart as we are to some extent. And I think that you're exactly right. Whatever we can do to hypothesize new chemistries, new structures, mechanisms to take advantage of, that's where we're at right now. We're right in the midst of what I think will be an exciting new time. This example here on PCSK9, work that was done at Merck during the time that I was there before I retired by a team of scientists that included colleagues at IRBM in Rome and Raw Pharma in Cambridge, Massachusetts. This is beautiful work to illustrate the interface of diversity using mRNA display to identify leads, as well as structure-based design over and over and over, iteratively to lead optimize, starting with a relatively weak macrocyclic originating from the work of Raw Pharma, but then really taking off significantly with the crystal structure of one of those early leads that were relatively weak binders, maybe in the micromolar range, but seeing how they were binding to PCSK9 supercharged the medicinal chemistry effort.
One small comment, which I think is important for the listeners. You mentioned it, but I just think that people should, really grasp the magnitude of what was done here. What you see here, the hit molecule from Ra, it's, I think, one of the first examples where you have completely novel and completely synthetic peptide that is actually active in doing very difficult inhibition of protein-protein interaction that was just impossible to do before these diversity technologies, mRNA display or even phage display came to be. So this is something that we're stuck in the first wave in history, zero chance or one in a few billion that you would actually stumble upon something like that to get a perspective on this. So this is, even though it was a very difficult and let's say a hit molecule, the achievement here is really interesting and I think is really important.
That's exactly right. And I'm really glad that you jumped in to make that comment because, when we look at the diverse library screening, most of the technologies have been binding to a target, using phage, using DNA-encoded libraries, using mRNA libraries. And I think that we've all kind of looked at the numbers. You can achieve millions, billions, trillions to identify through the process that each of these technologies have a slightly different approach. But to identify those binders that have the most affinity. Sometimes people think, well, you're going to find the nanomolar binders, maybe even better than nanomolar. But in some cases, it may not be that potent a binder you find, and maybe exactly for the reasons you mentioned, Immanuel, that the target themselves are the tough one. There was some people like to joke and call it like the billiard ball, some of these targets, because they're so, spherical almost. There's hardly really serious deep binding clefts or nooks and crannies that we typically want to be able to utilize peptide side chains or, even small molecules will look for specific in proteins as a major docking site.
I agree, Tomi, but I just have to say also, like, give another small comment regarding this, because I think this is such an important point. We are not going to cover like all the display technologies and diversity right now, but you touched on a very important limitation of the technique, of the technology. So it gives you a huge diversity, but the output is a binder which is not necessarily active, which is a great limitation. So finding the active molecule, the best active molecule, and not necessarily the best binder in cases like this, where your actual active molecule is probably not the first and not even in the first batch of peptide that are the best binders, is a great challenge, and is exactly the place where one of the great limitations of these technologies, which are extraordinary, but still have significant limitations when you're trying to apply them for drug discovery and development.
Yeah, I think that I would say that for the methods that have been developed here and the companies that have developed the platform technologies, it's been a lot of work. There's, as you said, there's a process, an iterative process of identifying sequences and what I call cousin analogs that would further verify that you have a lead series that has some structure activity or structure binding relationships. And I think probably everyone have found binders that didn't do anything for whatever reason. But in some cases, if you don't know where they're binding, that doesn't really help you to optimize. So getting a structure or getting some insight as to the peptide is binding, assumingly that most of them are yet orthosteric or competitive binders, whatever cognate ligand would otherwise bind to the target, that's really important. And some of that can be understood experimentally by looking at a competitive binding assay. But in other cases, we're just looking at the binding direct methods. And I think, speaking to the work from Merck on PCSK9, it was really the X-ray structure shown here in the bottom left. That was just one of many. There were ones earlier, there were ones later, but they really provide the molecular map, if you will, of how the peptide was binding. And it's an interesting one, just looking at the structure here. You see what is a highly modified tryptophan side chain and it's sticking into a deeper pocket on the PCSK9 from the peptide itself. And it's held in place by virtue of a cross-linking chemistry of the peptide coming to the indole ring. But these are what I would say the fun part of the science and how computational methods can be integrated. You may say, well, can we do a de novo? You just give us the active site and can we generate a novel de novo peptide that binds to the site? I say, why not? It's possible. And this is where sometimes where you say the rubber hits the road, now we have to do the chemistry to make those peptides if they're not exactly, off the shelf building blocks or chemistries. It's a cyclic process. It's iterative, but nothing I would say right now is more exciting than the ability to think outside the box with methods. And in the case of this work, I think it's important to talk about enthalpy and entropy. But it was interesting when some of the latter structures came out, including the one shown on the slide here. Fundamentally, increasing binding affinity, which we normally think is by iterative improvement in enthalpy, that wasn't necessarily what was obvious in comparing some of these highly constrained tricyclic peptides to their original predecessor, monocycles and bicycles. Mm-hmm. It was really the constraint of the peptide to hold, if you will, all the key pharmacophore groups that were involved in binding to the target PCSK9, holding them perfectly in place, if you will. And so this was a beautiful example. I've never seen quite the same, at least what comes to my mind, clear evidence of entropy being a driver of affinity. So I think that was the exciting outcome from this work as well.
I agree. I think it's a great lesson for peptide drug discovery in general. And I think that when looking at this really outstanding work and great achievement, I'm trying to think about the years went by and the resources starting from the hit to the lead molecule. And this is exactly what we are trying to impact in Pepticom, basically, is trying to get through design something closer to the lead molecule as soon as possible, even though reaching a lead like that would really, I think this is something that is well into the future. But even starting point, which is something in between, save probably a lot of time and resources. And I think this is a very exciting time in history where starting to become a possibility even. I mean, this is something that was complete science fiction a few years ago.
I completely agree. And I liked what you commented about the future and the history. We have past history, which I try to incorporate into this podcast, but also recent history, like the PCSK9 work shown here. And if we go to the next slide, I'd like to comment about the future because I look at now the timeline and the milestones to look forward to in terms of peptide drug discovery as the fourth wave, where we have AI and structure-enabled de novo peptide designs. This is it. This is where we're going. It's, futuristic or not. This is the beauty of being a scientist, right? We can reach for the future, make it happen, build the bridges, build the tools, and hopefully, build the collaborative network in order to achieve it with teams that have all the skill sets and working knowledge to do it. So I guess, from my perspective, I'm glad I didn't retire and just go sit in the hammock somewhere, but actually stay engaged and try to be effective as well as I can as an advisor and a consultant to biotech, and also to support the cause for you and Pepticom and for others I'm working with, and for the field. I have a lot of friends out there most of them are still younger than I am. Still got to keep working and pushing the wheel up the hill for their various efforts. I'm a big fan of many of those people out there, scientists, friends, all of you at Pepticom, to do this work. Whatever I can continue to do, I'm more than happy to do it.
I have to comment on this because when I see you and our CTO, Dr. Amit Michaeli, look at a design peptide in silico that actually has an activity, and we're trying to optimize it based on the knowledge that we have and the model that we have. And I see the excitement in your eyes and in Amit's eyes. I'm still like, I'm a bit of a scientist in my heart, even though I'm CEO. For me, I think it's a great achievement. And I think that the better the technology gets, this would has the potential to really impact human health and science. And I think it's a great time to do this work.
I agree, and it's a pleasure to talk about it too in an informal way, as we are here. So I hope the audience will have enjoyed listening to this podcast, one of a series, and we'll tune in again next time, as I think we're going to talk about intracellular targets next time, and then I think we're going to dive into the fourth wave. As you astutely stated a couple of minutes ago, leverages the diversity, the benefit of more knowledge of structures beyond the natural sequences, cognate peptides and proteins. It's all coming together. So it's very exciting times.
I completely agree. And before we wrap up, first of all, thank you. It was really, really interesting and educational and fun. I remember something you said in the beginning of the podcast, that you used to synthesize the building blocks, the amino acids, by yourself. We are sitting now in 2022. We have something like 400 different non-natural amino acids, and they are all like pre-made for us. Really, just looking at small fact is really mind-blowing, and just shows you the progress that was made in the last.
Well, and also your comment makes me want to definitely share and acknowledge all the contract research organizations and development organizations, the people that make all these reagents, make the building blocks, that make the peptides to support worldwide efforts, all the service companies, all the instrument making companies. We tend to be, at least I tend to always think about drug discovery from my own personal experience, and sometimes don't think about the fact that it wouldn't be possible if all, platform company or structure, I should say, framework wasn't there, that we can just send an email and say, hey, can you make these 10 peptides for us in the next few weeks? It can happen. All this can happen. And it is an exciting time to be still engaged, especially to work with brilliant people that I have the opportunity to do so now, and in a multidisciplinary way. There are times when I really just can't sleep because something exciting is going on, and I just want to see it reach fruition. I guess for those out there who are doing drug discovery, I wish you good luck, because we're all in this together. We're all going to make a difference in the world. That's our ultimate mission, right? To make a better world. And in this case, make a better world through peptide drugs. I guess that's my final comment, and I can't think of a better one, Immanuel.
Excellent. Thank you. So we'll wrap up, and we'll stay tuned to our next episode in this journey in understanding the world of peptide therapeutics and their discovery. Thank you very much, Tomi. It was a real pleasure, and we'll meet again soon.
Sounds good, and goodbye to all the audience.
Bye-bye. Goodbye.